Thank you very much. So I want to say a few words about this picture here. It's a seaweed farm. And it's where I like to walk, if possible, every morning, or sometimes every evening, because it helps get my 10,000 steps. And I've lost a lot of weight in Okinawa because of the diet and because of the exercise, and that's great. I was beginning, I knew something was wrong. My brother said, "You look fat, Tim." And I'm a lot less fat now. But, so Japanese seaweed farms, Okinawan seaweed farms. So our house is sort of where you're sitting, roughly speaking. So we don't actually see it because there's jungle in between. And there are signs in the jungle which says, "Beware of the poisonous snakes." But they only come out at night, so that's okay. And we don't go out at night. So in Osaka, there is a statue to a woman called Kathleen Mary Drew. How many people have heard of Kathleen Mary Drew? How many people have eaten sushi? Well, you owe it all to Kathleen Mary Drew. Kathleen Mary Drew has a festival on the 14th of April in Osaka. What did she do? Well, she was a lecturer in the botany department at the University of Manchester. In 1928, she got married and was obliged to resign her post. That's amazing, isn't it? I mean, that doesn't happen today. I don't think. Anyone had to resign because they got married? No. But she came back to work, having had her, I'm not quite sure whether she had children or, I presume she had children. Don't remember that bit. And she worked on the alternation of generation of a seaweed genus called Porphyra, which lived in North Wales. And what she discovered was that, yes, the seaweed was a seaweed. I mean, it's a sort of, nasty stuff slimy things that aren't good when you go bathing in the sea. But she realized that a thing which had been classified as a fungus growing on old oyster shells and clam shells was actually the alternate generation of this seaweed, and it had been completely misunderstood by everybody who'd worked on it. And so the seaweed give out sort of spores, and the spores settle on the shells, and they germinate and they burrow into the shells, thereby avoiding being eaten by fishes in the summer. Okay? And then they give rise to spores which germinate back into the seaweed. So once this was understood, it was really possible to have a totally rational, consistent harvest of the seaweed that gives rise to nori. Before then, it was terrible. This bizarre piece of work on the alternation of generation of an obscure species of seaweed actually turned into a multi-million, in fact, billion-dollar industry, which now employs something like 65,000 people. And all the sushi you eat is down to this one woman's work. And if that isn't the most wonderful example of how basic research gives rise to, useful things. I mean, you would have said, I don't know how many of you did, but we used to learn all this stuff, the alternation of generation in ferns and mosses. It was very difficult to understand. And why did they tell us that anyway? Because they don't really, nobody really understands it. But, that was all that was known, so you had to learn it. And without understanding it, very bad in my view. Anyway, so, yeah. So that's what's going on here, totally rational agriculture, or aquaculture, I suppose, one should say. So I shouldn't really be standing here because actually I am a living fossil. I gave up running a lab at the end of 2010. I nearly died from congestive heart failure. I'm only kept alive by drugs. Actually, I was very amused to discover I take warfarin because otherwise, my heart doesn't work properly, so there are stagnant pools, and then you get blood clots. They go to your brain, and you get a stroke, which is not a good thing to do. So I take warfarin. I was amazed. Do you know what warfarin stands for? I don't know why warfarin. It says Wisconsin Alumni Research Fund. Cows were dying in Wisconsin because they were eating this plant that caused them to, it was a vitamin K antagonist, and these guys figured it out. Another wonderful, wonderful story, actually, beautiful scientific detective work, and keeping me alive to this day. So it's great. So the story of my life is I grew up in Oxford. My dad's office was in that tower there. Here he is. He was an expert on medieval manuscripts. This medieval manuscript is written in Greek, as you can see if you look in the front row, I think. I've no idea what it is. His business was to help scholars in the Bodleian Library locate manuscripts all over the world. He had a very good knowledge of where things were, and he was one of those band of scholars who followed the evolution of knowledge by looking for transcriptional errors. We do that now with DNA sequences, but Dad did it with manuscripts. The scribes would make a mistake, and then the following people who copied it, of course, copied the mistake. So you could tell exactly which manuscripts they'd read and then who then read theirs, And it's like a family tree. Great stuff. And his great moment, his eureka moment, when he spotted that the marginal comments in some manuscript belonged to that of the poet Petrarch, because he knew what Petrarch's handwriting looked like. This is just unimaginable. That's what... Anyway, so I couldn't do Latin or Greek to save my life, so I had no idea really what he was up to. So luckily for me, I had a wonderful young German science teacher, and I looked forward to his lessons fantastically because they were fun. And they only happened once a week, and it was a welcome change from all this wretched Latin and Greek, and French wasn't much better either. History I never understood either. Maths I was useless at. The only exam I ever failed was something called additional O-level maths. Now, somebody said I should never tell people this because it encourages them to be non-mathematicians, but between you and me, biologists, adding and subtracting, knowing when to divide and when to multiply is quite important, but beyond that, you don't really need it. Mathematicians don't like that at all. They think maths is the queen. And of course, if you're a physicist, you have to be a mathematician. We'll come to that in a second. Anyway, so my first science, so I really wanted to be, and the thing is that I knew, Gert taught me that I should be a biologist because I did terribly well in a science exam when I was about 12 years old. I came out very near the top of the school, even though I wasn't near the top, I was sort of somewhere in the middle. And I hadn't done any work for this exam. It just sort of came naturally. I just knew, what the right answer was. So I knew I would be a biologist. I had a friend who was a very good physicist, and I knew I wasn't nearly up to him. So I would have loved to have been a physicist, but couldn't do it. Because my first scientific hero was actually Marie Curie, and her biography made a deep impression on me. She was one wonderful scientist and one determined young woman. Bloody hell. But I also liked, she had a characteristic which is described, I think this biography was written by her daughter. She's teaching a course in physics, elementary undergraduate physics or something like that, and she says, I've got this pot of water. What can I do to keep it warm? And her pupils say, well, I'd wrap it in straw, I'd surround it in bubble wrap or whatever, the early 20th century equivalents. And Marie Curie says, I'd start by putting the lid on. That's the mark of a really good scientist. Very simple, right? So, and then the next biographies I read was Alexander Fleming, the discoverer of penicillin, but not the developer of penicillin. He was a useless chemist, brilliant microbiologist, hopeless chemist. And this guy over on the right, who you may never have heard of, I actually met him just towards the very end of his life. He was unfortunately suffering from advanced Alzheimer's disease and couldn't really remember anything. Norman Heatley, he was the man who discovered how to get the penicillin out of the culture vessel, how to grow the penicillin so as to optimize the yield, how to assay the penicillin, all crucial things in the development of the drug. Chain worked out the structure of penicillin. Florey was the mastermind behind it. A very peculiar man, actually, if you read his biography. And I love that story. I had to open a building which was called the Heatley Building. So Heatley was there. His wife was called Mercy Heatley, and Ed Southern said to me, she should really have been called Take No Prisoners. She was a very ferocious woman. Very funny and fiercely protective of Norman Heatley and his reputation. It was wonderful. Anyway, so penicillin is a fantastic wonder drug, and this is a very interesting case, I think, of translational translational science, because I guess Florey was on the lookout for things that killed bacteria and didn't kill people. So they tried first lysozyme, and that was sort of okay. And in fact, Fleming had played with lysozyme. And then I think it was Chain who'd read Fleming's paper and said, why don't we get this? And somebody in the Department of Pathology at Oxford had actually bothered to write to Fleming to get the penicillin mold. And so they grew it, and they found indeed, it was wonderful at killing bacteria. And then the story I really like is one dark night, so Heatley had worked out how to make about 2 grams of very impure penicillin. It was probably no more than 1% pure, so dirty brown powder, extracted from hundreds of bedpans worth of penicillin mold. And he and Chain injected it into 2 healthy mice. They blew the whole lot on 2 healthy mice. And Florey was absolutely furious with them. How could you waste all this precious material? But of course, this was an absolutely key experiment because the 2 healthy mice lived. Nothing wrong with them at all. So they did the negative control before they tested it to see whether it would kill the bacteria in the mice. And that, again, is my kind of scientist. Absolutely the right thing to do. And then the rest is history. And it took these basically 3 people a very short space of time before they were producing the stuff in industrial quantities and saving hundreds of lives at the end of the Second World War. Again, well worth reading. There are several books which give account for this. So back to me and my evolution. The University of Oxford had things called extramural lectures, and I went to them. They were sort of happened, I think, at 8 o'clock on Tuesdays after supper. And the first one was on evolution. It celebrated the 100th anniversary of the publication of The Origin of Species, and so I learned about evolution. That was very interesting. Here's Darwin's famous first tree. Then the next one was on the biological effects of ionizing radiation. So that also made a deep impression on me and converted me to a CND supporter for quite a long time, because the effects of ionizing radiation, if you're not close enough to an atom bomb to be fried to a crisp, instead, all cell division stops in your body if you get a lethal dose of ionizing radiation. And the descriptions by the doctors in Hiroshima are absolutely, I have a book called Hiroshima Diary, and it's unreadable because it's so harrowing. It's just absolutely ghastly what they had to put up with. But at that time, in 1960, nobody knew how it worked. A lot of people thought it worked on the membranes, the ionizing radiation, and it was quite controversial. Now we know it's almost all due to DNA damage, and I work with lots of colleagues who do that kind of stuff. So that was very, very, very handy. And then finally, last one was by a guy from the biochemistry department. Now, I mean, the truth of the matter is, this is the Boehringer metabolic wall chart, and it is deadly dull. And if you have to learn this stuff, it's just awful. And when I was an undergraduate, you had to learn this stuff, And what is the evidence for the Krebs cycle? That was a very popular question that you had to answer. I now can't remember what the evidence for the Krebs cycle is, and it doesn't really matter. But I mean, the Krebs cycle hasn't gone away. Let's get this straight. But the thing that puzzled me about all of this, I was very good at chemistry. I had a wonderful chemistry teacher at my subsequent school. So combining biology with chemistry was a really logical thing to do, and I was terribly lucky because you were sort of getting on the ground floor of molecular biology, actually. And the thing that worried me was there were all these pathways had been worked out, but nothing was known about the control of those pathways, and it seemed obvious to me that there must be something controlling them, and that sort of fell out while I was an undergraduate, and that was very exciting. So I went up to Cambridge to learn how to be a scientist, and this is my college. This is King's College Chapel. later as a research fellow, I had a room up there, wonderful view, one of the best views I've ever had in my entire life. I lived there for about 7 years. So Cambridge is a famous place for science because, starting with the great Newton, nature and nature's laws lay hid in night. God said, "Let Newton be," and all was light. I love that. Alexander Pope composed this couplet as a, it was going to be on Newton's tomb. I don't think it ever made it to the tomb. But so there, this, you know, you were already in a grand tradition. But then, much more recently, in the middle of the 19th century, Maxwell, who was a brilliant mathematician, and unfortunately, A-level physics stopped well short of Maxwell because understanding electromagnetism actually requires rather a high degree of mathematical skill, well beyond me. And then, you know, all this was done in an area not much bigger than this campus, in fact, far smaller. You know, within 50 yards, these amazing discoveries had been made. I mean, you know, basically the nature of the atomic nucleus was discovered, then the proton, the neutron, the first splitting of the atom, and then their successors, also in the physics department, Watson and Crick and Kendra and Perutz. So, and I used to walk past this place on the way to work, and you know, if you were sort of feeling good about yourself, you'd say, gosh, this is amazing. I'm in this grand tradition myself. There's nothing stopping me from being a brilliant scientist and making fantastic discoveries. But of course, normally things weren't going so well, and then you thought, oh shit, this is terrible. You know? I can't possibly measure up to these guys because they're so brilliant, you know, and there's no way. I'm just an ant in the grand scheme of things. So there they are. You know, I was very lucky. I knew Francis quite well. You know, occasionally he would come to seminars and I'd sit next to him at dinner. And I never had a conversation with him, but that he illuminated what I was doing. He was so intelligent, so clear. Again, simple and clear. And he was the only person really in Cambridge who understood all the physics and all the chemistry and all the biology. So my generation were absolutely in awe of this man. And then the silly bugger went off to California and started working on consciousness. And I don't think he ever really made much advance of that. So I think that's a bit of a waste, really. Jim, on the other hand, who I also know quite well, he was very kind to me once by sort of generously donating a tiny fraction of his royalties to help pay for some work that we did on molecular biology of the cell and the problems. And he's a very strange character, as anybody, I don't know how many of you heard him talk, but he's a little bit crazy. But also, you know, I like him, actually. I like him. He gets a bad press. And Kendra I never met. But of course he was here in Heidelberg as the director of the founding director of EMBL. But I show you this picture, which I love. You know, here he is. This is a BBC TV science program, and this is the sausage model of myoglobin. It's a very early model. And I show it because you can see the heme lurking in the heme pocket of the myoglobin. As you know, hemoglobin has 4 chains, and myoglobin only has 1. Okay, so why do I show you this? I don't know why I show you this, actually. I know why I show you this. There's one I'm not used to. I love this photograph. The ash is practically dropping off the... Anyway, so Mono came in 1962 and gave 2 absolutely fantastic eye-opening lectures about the control of gene activity. And I went to my supervisor and said, you know, is this true? Because, you know, if it's true, it's fantastic. So Asher said, bit early to say, you know, but this was the year they won the Nobel Prize. So it just proved that not everybody in Cambridge had read the literature. And it was true that most of the literature in this case was in French. So perhaps there were excuses. But it was a real eye-opener. I mean, and again, brilliant, clear, logical work. So then the question was, I did well enough in the exam, so I was accepted as a research student. And I should say in those days, I think I'm right in saying that me and my contemporaries, we all wanted to do research. We had no idea what research really was. We had a sort of vague sense that you needed to be pretty dedicated and that you might not earn a lot of money. And so I don't know where we got the confidence to do it. I mean, it was, in a way, you could say we were really stupid, but you know, that's what we wanted to do. or at least that's what we said we wanted to do. And actually all the three people who I remember saying this, we were asked by a supervisor in first supervision, they all turned into researchers. So the problem with being a researcher, of course, is what do you work on? So I went to my supervisor, who was the same man who hadn't known about the work of Jacob and Monod, and said, Asher, what shall I work on? And he said, well, why don't you go to the library and find yourself a problem? So I went to the library, and I found a terrific problem, which, as far as I know, has not been solved to this day. And it involved making rats nephrotic. That means making autoantibodies against their glomerular membrane so that they pee out albumin. And the observation was that when the rats pee out albumin, the liver upregulates albumin synthesis. And that's what I wanted to work on. So how to do this? Well, you first had to kill the rat to get its kidneys out, and then you had to mash them up and inject them into a new rat. And I discovered I hated killing rats, and the rats hated being killed by me. And it was a sort of and luckily for me, I hadn't read the materials and methods properly, so I used the wrong kind of rat. And they did not get nephrotic when injected with their own kidney extract. So, you know, I had these rats in a cage, and I was trying to look for proteinuria, you know, awful. So about this meeting was actually in 1965, and in April, I think, if I remember right. We went to my first scientific meeting, and this was an absolute eye-opener. It was about hemoglobin synthesis and structure, because I guess Kendra and Perutz had just worked out the structure, so they had a lot to say. But these two talks were about hemoglobin synthesis, and Henry Borsuk had worked on rabbit reticulocytes, and he'd found that if you starved them of iron, they stopped making the protein. So that was very interesting. He also had worked on sea urchin eggs and compared, bizarrely, red cells with sea urchin eggs. They really don't have very much in common. I mean, you know, sea urchin eggs are at the beginning of their life, red cells are at the end of their life. I think the only thing they have in common is they're both round. But anyway, they sort of, you know, bear that in mind, because this sea urchin business is going to come in. But the talk that gave me my PhD project was by Vernon Ingram, the discoverer of the flaw in sickle cell anemia. And Vernon was also working on the question of how heme regulated protein synthesis. And the theory was this. So ribosomes get onto the globin messenger RNA, and the chain comes out longer and longer. And at some point, it forms the heme pocket where the heme is going to bind. And what he said was that if there wasn't any heme around, the ribosomes stopped when they got to the place that the heme should go in. How the ribosome knew how to stop or anything like that, completely obscure, but the evidence he presented suggested this. However, when we went back to the lab, I was rather excited by this. What a wonderful feedback mechanism. Gosh, phew. I wonder how it works. But actually, he got it completely wrong. His evidence, if anything, said exactly the opposite. So we thought, and this is the step I can't reconstruct, we thought, well, why don't we do the experiment ourselves and try and find out what the truth of the matter is? And, I mean, that seems an extraordinary act of foolhardiness viewed in retrospect, but that is exactly what we did. And when I say we, it was a mixture of Lou Reichart, who was my lab partner for the first year, and then subsequently Tony Hunter joined us. And Lou knew how to make these rabbit reticulocytes, and we could label them, and we could work out actually better ways than Ingram had used, to look for cues, and we found that actually there weren't any cues. If we formed cues deliberately by various tricks, we could detect the cues by our method. So everything was cool. And so Tony and I sort of did our PhD together. Here, Tony is eluting radioactive peptides, and we sort of a variant on the Dintzis experiment for those of you with long memories. And that was very good. And along the way, I went to my second scientific meeting, which was also tremendously influential, because I ran into this guy here. It was in northern Greece, and I got on terribly well with Irving. And by this time, this is the sort of the paradigm. You know, you add heme and globin synthesis goes on. You don't add heme and globin synthesis starts off all right and then stops, or almost 5-10%, something like that. If you add back heme later, then protein synthesis recovers. And by the time I got as a postdoc I actually spent a sabbatical as a graduate student, which is rather unconventional, but it was good fun, and I learned about American labs. And I learned a lot about collaboration, actually, and the importance of sort of bringing different brains to bear. I really enjoyed it very, very much, the open atmosphere of this American lab. And he was a medic, you know, he was in charge of he was the boss of if you mentioned his name, people quaked because he was very powerful. But to me, he was just a pal interested in the same project. So there's a sort of little lesson there, much older than me, you know, 20, 30 years old or something like that. But we got on very well. And then along the line, we worked out how to make a cell-free system that would do this. And that was a huge advance because it meant you could do an experiment any time of any day you wanted. You just freeze the stuff in liquid nitrogen and take out a tube, you can do it. But the only trouble was you kept on doing this experiment, and that was very reproducible, but it doesn't tell you how the control works. And it was a long time, it turned out, before we figured that out. So along the way, I got rather depressed and started to do sort of naughty side projects. I had a technician who was more or less twice my age. She was called Grace. I loved her. But she went home at about 5:30, and then I would start a whole new project after she'd gone home. And the project was to map the order of genes in poliovirus. And I discovered that poliovirus wasn't translated by rabbit reticulocyte lysate. So I deduced that there must be factors in HeLa cells that promoted the translation of poliovirus, because HeLa cells get infected by poliovirus and red cells don't. Perfectly correct logic, absolutely confirmed up to the hilt many years later. So I went to Ellie and said, can I have some poliovirus-infected cytoplasm? And she made some, and we added it in, and to our astonishment, it shut off protein synthesis. Both these things have heme now, but when you added the poliovirus-infected cytoplasm, protein synthesis shut off just as if you'd left out the heme. So there could have been a heme chelating agent in there, I suppose, but we didn't think that was very likely. And in fact, it turned out that the inhibitor was very large. Not only was it very large, but it also resisted digestion by DNase, by RNase, and by protease. Oh my God, it must be a complicated, carbohydrate. Our hearts sank. And it was a long time. Interesting, I say in time of war, because this was the moment when Nixon was invading Cambodia, and many of my friends were in danger of being drafted. So it was an awful feeling. It's a little bit like Brexit, actually, that the whole section of the population wants to do something that you consider profoundly wrong. In fact, and evil in the case of Nixon. Well, I think Brexit is pretty evil, actually, but that's another, let's not go there. So anyway, it took us a while to figure out, and it sort of felt almost wrong to be doing these experiments. We were on the track of finding out what this stuff was, while these crimes were being committed on our name. And the whole atmosphere was incredibly polarized, very interesting, actually. I've never known really, maybe we're getting back to that stage, politics sort of intrudes on everyday life. Everybody has a view. Anyway, it turned out that in the end it was double-stranded RNA, which, as you know, resists digestion by ribonuclease. And I had to go to Jerry Hurwitz and say, what kind of an enzyme would digest double-stranded RNA? And he said, micrococcal nuclease, and don't forget to add the calcium. So that's how I discovered that you could, that micrococcal nuclease had absolutely zero activity if you didn't add the calcium. And that led to the development of the messenger RNA translation system that Hugh Pelham eventually developed. And then in collaboration with another woman, Nechama Kosawa, I discovered that tiny traces of oxidized glutathione caused exactly the same kind of defect. That was very weird. So we've now got three quite different things: lack of heme, presence of tiny amounts of double-stranded RNA, presence of tiny amounts of very mild oxidizing agents, all do the same thing. And what they do, I didn't figure out until I went back to Cambridge. I took a 5-fold cut in salary. I went from earning $11,500 a year to earning £800 a year, which was barely enough to live on. But that was okay, because I worked with this man here, Richard Jackson, who was a university teacher and a lot cleverer than I was. And we made a very good team, because he would tend to get sort of bogged down in detail, and I liked broad brush. So he restrained my wilder enthusiasms, and I sort of pulled him forward when he got stuck in detail. And perhaps this is a good point to say where, in my experience, my happiest times have been always in collaboration. Always in collaboration. Never, I hate working by myself, but never with more than 2 or 3 people, really. I mean, when I had a lab of about 10 people, it was very difficult. I find that hard to handle. But Richard and I and a couple of graduate students were, a very tightly knit team, and it was great. So what was happening? Well, it became immediately clear when we started running sucrose gradients, and we were very good at that. I've been doing that for years and years and years. And there was this funny peak that people had noticed but not paid any attention to of methionine labeling over the 40S, the small ribosomal subunit. And what we discovered was when you left out the heme or added double-stranded RNA, this peak went away. And that peak went away before the polysomes went away. So it looked very much as though it was a precursor. And what we found was that actually this was initiated tRNA, which was bound to the 40S ribosomes before they bound to messenger RNA. So this was a real paradigm shift. People would tell you, tRNA only binds to ribosomes as instructed by messenger RNA, but here was a case where actually it was the tRNA that bound first and went looking for the start of the message. So we had a terrible time with the reviewers, but we were right. And that's always a good position to be in when you're confronted with hostile reviewers. So, we were rigorously defended. So that was great. So then that really enabled a great breakthrough, except that at that point the lab burned down. And it turned out that was the best thing that could ever have possibly happened to her. Is this a challenge or an opportunity? Well, it was a wonderful opportunity. Why? Well, because, of course, we had to move to a new lab. And also because all the old data, actually at that point we were terribly baffled and puzzled, very foggy about what was going on. Lots of funny experiments that didn't really make sense. They were all burned up. So, it was all like making a wonderful confession in the Catholic Church. You started with a clean slate. But more importantly, I'll show you in a second where we went. Maybe here, yes. Here. So we were here in the hospital in a hematology teaching lab, and there's the famous laboratory of molecular biology, absolutely loaded with Nobel Laureates. And Max Perutz was very kind to us. He said that we could use his stores and have lunch in his canteen, and which Mrs. Perutz ran at the time. And there was a wonderful insurance thing. So in 6 weeks, we had everything. The last thing to come was a pH meter, I remember. I don't know, the radiometer pH meter took ages to come. And you actually can't do anything until you have your pH meter, if you think about it. Can't make up any solutions. So we had a sort of, maybe that helped too. We sort of thought about things a bit more. Anyway, but the key thing was the lunchroom. Because you'd sit down and have lunch, and Francis Crick would come up and explain about nucleosomes, and that was pretty exciting. And he was working with Roger Kornberg, who later won a Nobel Prize, not for that, but for studies on transcription. And there were all these people. I mean, I was terrified of Sydney. Cesar was really sweet and lovely, and Aaron had a graduate student with whom I collaborated. I was most friendly with John Gurdon and his lab. So these are pretty classy scientists, actually. And I think, again, Jim said that if you want to get ahead, you should work with people who are better than you are. I mean, this is a really good advice. If you never find people who are really good, it's nice to be in their company. But we weren't really working with any of these, except for perhaps the collaboration with Aaron's graduate student, David Zimmern. And, these are all Nobel laureates, and they're all my contemporaries. I mean, I think the interesting thing is we would not have considered, I certainly didn't consider myself in the same league as these great pioneers or anything like, right? Because they did really heroic things. And we were just sort of following our noses. We had interesting problems. We would discuss them with each other. And, people would make suggestions, keep you on the right road, keep you honest. And it was terrific. And the result was that because of all this combination of things, burning the lab down led us instantly to the answer. And the answer was that double-stranded RNA or lack of heme or presence of mild oxidizing agents activated a protein kinase which phosphorylated the initiation factor that bound transfer RNA to the 40S subunit. Problem solved. And in retrospect, one said, oh, God, it's so simple. Why the hell didn't we think of that before? And I don't know, but you never do. Nowadays, this is a structure, I guess, of probably an 80S ribosome. There's the messenger RNA, and there's the transfer RNA. Here's the EIF2, which gets phosphorylated. And actually, it turned out that what phosphorylation does, the first operation, it can carry in the first tRNA, but it can't recycle. That's what gets blocked. And that had us foxed for a long time. But basically, this is an important thing. We had a problem. It had taken me maybe 7 years to solve it with the help of my friends. And by the way, beating out the competition. Irving London was working on this still. And we got there first, and that was very satisfying. I think competition is good if you win. And now it turns out, this is actually very interesting. This is, again, sort of translational research in more than one way. So it turns out there are 4 of these enzymes. Now they've been cloned, and thank God I didn't stick with it. It was a very long time before they were purified, cloned, and sequenced. So there's protein kinase R, that's the double-stranded RNA. GCN2, which we didn't come across, that's responsive to amino acid starvation, as the name implies, was found in yeast. The heme control kinase. And another one called PERK, which is involved in the unfolded protein response. And I discovered recently, it was actually Richard Jackson told me, that actually PERK inhibitors show promise in the treatment of neurodegenerative diseases, because what happens in the nerve cells, apparently, is that these proteins get misfolded. They trigger the unfolded protein response, and that shuts off protein synthesis in those cells. And it's the shutting off of protein synthesis that causes the nerve cells to die. So if you inhibit that enzyme so they don't shut off protein synthesis, the nerve cells don't die, and you don't lose your memory. Now, I don't know whether these things are actually working or whether this is just pie-in-the-sky wishful thinking, but it's sort of, I would love to say, well, actually, the research I did led to the cure for Alzheimer's disease. That would be wonderful. So, as you know, phosphorylation controls lots of things. Here's a good example of a dead enzyme on the left and an active enzyme on the right. And you can see that actually the changes in structure are really pretty subtle as a result of phosphorylation. And as you also know, phosphorylation sometimes inhibits enzymes, sometimes it causes complexes to form, sometimes it blows preformed complexes apart. You just never know until you look, but it's very widely used. So we were pleased with ourselves for having done this, and I organized a meeting, and one of the people we invited to the meeting was Tom Humphries, because he worked on the control of protein synthesis in sea urchin eggs. Because I was already looking, having solved the problem, what do you work on now? Well, you can go into more detail and all kinds of things, but I thought, it's time to find a new problem, at least as a sort of side project. So we invited Tom, and we became friends because, unbeknownst to me, he was a keen cyclist and wanted to go for a cycle ride. So I lent him my bicycle. And everything came from just lending him my bicycle, actually. We became friends because of that. Don't neglect to be kind to people. You never know what the consequence. So I had no idea, but it turned out that Tom was the course director in Woods Hole. And he said, why don't you come and help teach the embryology course next year, and maybe we can do some experiments on sea urchin eggs. So Woods Hole I knew already because Irving London had a house somewhere about here. And I knew it was beautiful, and you walk down the main street. I remember running into Sidney Brenner in the local supermarket. There was only one supermarket. It was very small, mostly a sandwich store, actually. And it was like that, famous scientists walking up and down the street. And wonderful lectures all the time, because people would come through on the way to going to Gordon conferences and things. So fantastic education, actually. I learned all my cell biology in those summers that I spent teaching, and lots of discussions too, so you could really get to the bottom of things. I liked that a lot. The first year I was there, I did absolutely bugger all, because it turned out they were incredibly badly equipped. They didn't have any gel apparatuses. So the next year I went back in '79, and I knew what to bring, so I brought with me my own gel apparatus and Eppendorf tubes and tips and peristaltic pumps. My suitcase was absolutely... And in those days, believe it or not, you could actually take a dry ice box, a styrofoam box full of dry ice with rabbit reticulocyte lysate and tobacco mosaic virus. You didn't declare the tobacco mosaic virus. They wouldn't. And you could put it in the overhead compartment of... And it was great. It would survive the transatlantic trip, and you'd get it and put it into a Revco the other end, and all was well. So that's how I used to operate. And you couldn't do that nowadays. I don't know what you'd have to do. You'd probably have to sign millions of forms, and they wouldn't allow you to do it at all. They think you were a terrorist. So that was great, and you could indeed get sea urchin eggs. You give them a 12-volt alternating current electric shock, and the females shed their eggs, and the males shed their sperm, and if you mix the eggs with the sperm, they get fertilized, and they turn on protein synthesis. Great. So what's happening here? Well, I assumed, of course, that it was some sort of phosphorylation change at this time, and we looked for that. And we found, indeed, that the 40S ribosomal subunits got phosphorylated, but that actually that wasn't what turned on protein synthesis. So that was a little bit of a bust. But we did learn how to fertilize sea urchin eggs and how to run good gels. And that wasn't trivial because most of you probably don't know this, but there's something in a sperm called the acrosome. And the acrosome is full of proteolytic enzymes, which it uses, I think, to burrow through the outer layer of the egg when it fertilizes it. And if you simply add SDS to fertilized eggs, what happens is that you release all these acrosomal enzymes, and they're the last ones to get denatured. Meanwhile, you've denatured all their substrates, so you get a phenomenon that we used to call upper gel wipeout. All the big proteins were cut in half by these proteases, and we had to work out ways of inhibiting them. That turned out very simple. Trichloroacetic acid was the answer. Not too sophisticated, but these things had to be worked out. But along the way, I also discovered, this is an oocyte, by the way. These are eggs, and I will fertilize them. There we go. And as well as turning on protein synthesis, they start dividing. And they divide incredibly synchronously, naturally incredibly synchronously. So that's very important. And in the summer of '79, John Gerhardt, an expert in enzyme regulation, came and gave a talk, which I found absolutely fascinating, very stimulating. And he talked about frog oocyte maturation. Oocytes become eggs, and they have to undergo a process called maturation, which actually involves two meiotic divisions. In frogs, this is triggered by progesterone. So if we add some progesterone to these eggs, watch carefully, a white spot forms, and then they do the first meiotic division. That little is the first meiosis. Okay, you can see that rather more clearly here and in the following slide. So these are starfish. You can't see into, you have no idea what's going on inside the frog oocyte. Starfish are nice and transparent, so let's add their hormone. The nucleus just dissolves, and then, and you see there and there and there and there. That's this tiny little cell which contains half the DNA of the oocyte. And these movies were made just up the hill, by the way, by a friend of mine, Peter Leonard. This is great. I love this one. So the chromosomes line up on the spindle, and then they do anaphase, and throw away half the DNA. Great. Okay. So Gerhardt described these amazing experiments done by Yoshio Mazui in Yale. He sucked out the cytoplasm from a mature egg, which was arrested in second meiotic metaphase, injected them into a fresh oocyte, and the fresh oocyte responded very rapidly as though it had seen progesterone, and this conversion did not require new protein synthesis. So it turned out that what progesterone did was to activate an inactive precursor substance, which was called maturation promoting factor, and it was protease sensitive and heat labile, so apparently an enzyme. And I thought, wow, an enzyme that catalyzes a cell cycle transition. That's amazing. Enzymes, as I've explained already, do usually very boring things like catalyze the Krebs cycle. But this was a really interesting thing. Catalyzing a cell cycle transition, what could it possibly be, and how does it possibly do it? And I thought, what a delicious problem. How I wish I could work on that. But I wasn't. But I began to be aware of the connection between protein synthesis and cell division. And I wondered sort of vaguely whether sea urchin eggs had MPF, and we discussed that with Teddy Salmon, whether he was teaching himself how to micro-inject sea urchin eggs. And we never got around to it, but we were sort of thinking about this. So the only thing you could do, you couldn't find out what it was because it was terribly unstable. If you tried to purify it, I mean, simply diluting it to put it on a column, it just disappeared, and nobody knew why. But you could assay it, and it was always high in metaphase cells and always low in interphase cells. And it wasn't just in frogs, but in starfish and even in human cells too. So, and human MPF will make frog eggs mature. So there was a sort of interest. There was a it became, people gradually began to focus on what this was. And then by 1982, the actual control of protein synthesis experiments were going really badly. And I really had no idea. And I just sort of was idly wondering. And I was reading this book looking for clues, Artificial Parthenogenesis and Fertilization, mostly about sea urchin, virgin birth in sea urchin eggs. Okay? And the question was, do parthenogenetically activated eggs make the same proteins after their activation as properly fertilized eggs? And I think I was sort of prompted to do that because my parents were both very devout Christians, and I had been brought up to believe that the virgin birth of Christ was a dogmatic article of faith, which by this time I no longer believed. But the idea that one could actually do molecular experiments on a dogmatic article of faith I found really rather attractive. Here we are, the Holy Spirit, whooshed. What was it? What could it have been? DNA, RNA, protein? This is a wonderful picture. Poor old Adam and Eve being expelled from the Garden of Eden. This is sort of making it all right. I've gotten very interested. I started reading St. Augustine. Actually, really, I went to Serbia shortly after going to Croatia. I never understood, the Croatians are Catholics and the Serbians are Orthodox. So what is the difference between Catholicism and Eastern Orthodoxy? Well, one thing is beards. Believe it or not. So the Greeks thought that beards were a sign of manliness, whereas the Romans thought that beards were a sign of effeminacy. So Eastern priests all have long beards, and Catholic priests are all clean-shaven. And this was a tremendous source of friction between. And I learned about the great schism of 1054, when the churches sort of excommunicated each other. And I discovered that my godfather, who was a very distinguished medieval historian, had actually written a history of the church. It was absolutely fascinating. And the arguments about the nature of the Trinity, is there one God or are there three gods or one in three? I mean, again, people were killed because of their beliefs about this stuff. It's very interesting. It's like scientific controversies, only about religion, It's no different, really. Anyway, so I did this experiment, and this is what I saw. It had nothing to do with parthenogenesis, but everything to do with properly fertilized eggs. So I saw this protein here, and it was the strongest protein at early times, and then it went away, and then it came back again, and then it went away again. And I could show that every time the cells divided, this protein went away about 5 minutes before you could see they actually divided. And I thought, oh my God. Obviously, we know that inhibiting protein synthesis blocks cell division. So we're actually looking, I actually did not dare to have this hypothesis. We're looking, MPF is synthesized and then it's degraded. Because, of course, at the time, this was absolutely impossible, completely and utterly impossible. Proteins could not be degraded like that. There was no known mechanism for getting rid of one protein, whereas all the other proteins, we know about proteases in your stomach, but they digest everything. Here was one protein going away, and because it was so impossible, nobody had even suggested it theoretically. And I think that's why the Nobel Committee sort of liked it. I mean, I think I was just incredibly lucky, really, to see this. And I was the first person to see it, and the first person also to correctly interpret it. But I think you'll agree, you don't need to be a genius to interpret this. I don't think I have a huge intellect or anything like that. It was pretty bloody obvious, So I sort of realized that, this was going to be the life's work, and it was likely that this would sort of last me for the rest of my career, which was a very happy thought, because actually changing fields is quite uncomfortable. I don't know if you've done that, because you go from being an expert to being like it was when you were first a graduate student. You know absolutely nothing. You know absolutely nothing. You can't even understand what your lab mates are saying. It's just awful. And then we looked in other organisms. So this turned out to be a bit wider. And I was very pleased. In clams, there are 2 of these things, which we'd been studying already with my friend Joan Ruderman and her graduate student Eric Rosenthal as translationally regulated proteins. But Eric had never noticed that they went away because he'd never done this really simple experiment. Okay? Notice that the yield was sometimes rather variable, but never done this really simple experiment of taking time points. And I was just in the habit of taking time points. And if you're waiting to look at a pattern, it's more interesting to take occasion. It gives a certain robustness to the experiment, too. So, anybody could have done this experiment in the preceding 10 years, and they didn't. And there were reasons why they didn't. So I realized that I had to figure out what these funny disappearing proteins were. And I realized I needed to learn about what was known about the cell cycle. And the answer was, in those days, very little. And, there was M phase and S phase, and M phase was kind of complicated. I mean, it's a bit hard to say, what do you mean catalyzing entry into M phase? Does that mean catalyzing the breakdown of the nuclear envelope, or the condensation of the chromosomes, the splitting of the centromeres? And then there's the metaphase to anaphase transition and what's going on there. When we measured the disappearance of cyclins, it turned out the cyclins went away about 90 seconds before the chromosomes came apart. So, they looked like there might be, but we had no way of inhibiting it. So, and, if you read about reviews of mitosis, it just looked horribly complicated. So it didn't really seem that the simple idea you make something and you destroy something could possibly apply, because it's much more complicated than that. But, Lee Hartwell's experiments had a certain logic to them, and he was very logical and ordered these genes. He didn't know what they were because he was always the pioneer. So, when you're the first person with a gene, there's nothing to compare it with. So he had no idea what these things did, any of them. Poor man. But also, it was interesting talking to him because I always wanted to know how things worked. I was really at heart a physiologist. And Lee was just sort of a logical geneticist. It was really interesting. He didn't have much interest in how these things what they actually did. So I found that curious. But, it takes, again, a sort of lesson here is it takes all sorts to solve scientific problems. It helps to come at them from different angles, I think. And then Paul got going on this, and for some reason this slide is awfully washed out. And he, in trying to enrich for large cells, he actually enriched for small cells because the small cells clump together. And he thought, wow, this is amazing. This is a temperature-sensitive mutant that accelerates the cell cycle. And he looked for other mutations and found that CDC2 had an allele which also accelerated the cell cycle, although a recessive allele block. So, if you knock it out, the cells can't divide. So. And that appeared to act in Paul's yeast here, whereas CDC28 was catalyzing start and appeared to act here. But when they were cloned and sequenced, and you could begin to try what happened if you express CDC28 in fission yeast or CDC2 in budding yeast, it turned out they were interchangeable. And they both control both of these things, and you can either kill them or make them. And when they were sequenced, they were found to encode a protein kinase, but the protein kinase had absolutely no activity. And again, it never occurred to us that this protein kinase might require an activating subunit. Actually, I meant to put in a slide here to show how we did find that out. Jim Mahler and Fred Locher eventually succeeded in purifying MPF by changing the assay a little bit. And it had 2 subunits. And I told Jim, as soon as we have antibodies against cyclin, we'll check whether the large subunit is cyclin, because the small subunit was clearly CDC2, and it had protein kinase activity. So that was basically how we did it. And when I say we, it was John and Jeremy. John was the first person to clone sea urchin cyclin B. Jeremy was the first person to get frogcyclins. And I was absolutely thrilled when we found frogcyclins, because then one absolutely knew they were going to be in humans. A frog is a human, basically, to me. So they were great students, and again, absolutely wonderful. I loved working with these two boys. They were so fun, it was absolutely great. And I also used to, every time I passed through London, I used to call in on Paul, and we would discuss. He would tell me what he was up to. I would tell him what, and I would ask him, have you seen a cyclin in your thing? And he said, no. Anyway, so it turned out this was the answer, that cyclin is the activating subunit of CDC2. Very simple. So when you understand what had been completely mysterious was that the amazing truth was that cyclin builds up, turns on CDC2, and the cell enters mitosis by phosphorylating hundreds of proteins. And then cyclin is destroyed, so are one or two other proteins. There's a very good system for getting rid of proteins, and as you know, it's quite complicated. CDC2 turns off. Proteases get activated, and the cell exits mitosis. So then everyone said, oh, wow, you understand the cell cycle, so now you can cure cancer. But I remember the atom bomb experiment. What happens if you inhibit protein synthesis? You die. So was there any evidence that cancer cells have different cell cycles? Not really. But what we did find was that there were lots of different kinds of cyclins. So the ones I was interested in was catalyzed entry and mitosis. Cyclin E, CDK2, catalyzes entry into S phase. And you see, it's interesting, these are different enzymes. They need to be different enzymes, because if you turned on these guys here, you would immediately enter mitosis prematurely and skip S phase, and that would be no good. So cyclin E, CDK2 is a kind of wimpy kinase that just does this business and can't initiate it. And then Chuck Scher and David Beach discovered cyclin D and CDK4 and 6, its partners. And these were clearly important for actually getting out of G1 and into S phase, passing the restriction point, very specific for retinoblastoma protein and its methods. So is cancer a disease of the cell cycle? Here is a man with cancer that I photographed in the St. Bartholomew's Medical Museum. And you can see that something is horribly wrong with his lip, and those cells are dividing out of control. You can also see that those cells are awfully good at getting a blood supply to themselves, which helps them to grow. And, how could we stop them growing? And most of the cells of his chin, are perfectly well behaved and still remember that they're chin. So what the hell do these cells think they're doing? And, you've probably all seen Hanahan and Weinberg's. This is the original thing, and it's bloody complicated. And the question is, we've been talking about this the cell cycle engine over here, but really it's all these things on the outside that are turning on the cycle that are more interesting, because there's clearly nothing wrong with the cell cycle in cancer cells. If there was something wrong, they wouldn't be able to divide and they wouldn't be cancers. And then there's this interesting thing called the abnormality sensor. I've always wondered what the abnormality sensor was and what is abnormal for a cell, and I think that's still interesting. So, but recently there's been a bit of a breakthrough, because we know it's a lot more complicated, this, and cyclin D, CDK4, there it is, phosphorylating RB, which is an inhibitor, so it removes the brakes on E2F, so that allows you to move forward past the restriction point, and then there are inhibitors up here. It's quite complicated. But it looks like, growth factors work through cyclin D, CDK4. So the drug companies had invented some rather selective cyclin D, CDK4 inhibitors. And Slayman and his team just grew, hundreds of cell lines to see whether any of them were susceptible to these inhibitors. And they found that a particular class, luminal estrogen receptor positive human breast cancer cell lines, were exquisitely sensitive to these inhibitors. So then they started using them in human beings, and indeed, this stuff, palbociclib, seemed to have great promise in these breast cancers. But it's sort of, what's interesting, that's great if we actually, some of these cyclin kinase inhibitors are working, but it doesn't make any sense to me, actually. They've got no right to work, actually. It's all backwards. So I was thinking about this last night, and I wonder whether this isn't actually an off-target effect on some other kinase that they don't really know about. And it seems to me entirely possible. I mean, it may be because they inhibit cyclin D, but they haven't actually proven it yet. It just works in treating cancer. I guess that's the main thing. So this is still an active, very interesting area of research, and hopefully it will continue to go forward. So I don't know. I hope I've made it clear that the road to discovery is a sort of rocky one, and you never know where things are going to lead, and you often get inspiration from very funny places that you never expected, and you've just got to keep your eyes open. People say, what would your advice be to young scientists? And I tell them to keep their eyes on the horizon, but their feet on the ground. It's terribly important when you take a step to make sure that then what's going to be your back foot is really firmly planted before you take the next one. Otherwise, you fall over and crumple in a heap. And the other thing I think is, don't be frightened of new things. Go for it. If you've got a good problem and you need to learn something you don't feel very comfortable with, that's absolutely fine. Just go to a lab where they know how to do it and learn. It takes about a week usually. Right? It's sort of surprisingly easy, yet terribly, terribly difficult. And, looking at this sea of the faces, you're the future. It's up to you. Good luck.
Thank you very much for a wonderful, inspiring presentation. I think we have time for a few questions.
I hope so, if anybody has any. I usually stun people into silence, but but then we need a sort of warm-up man.
Yeah, just starting with the first one. So what could be the off-target inhibition? We have our own little CDK4/6 inhibitor project in MRL.
Oh, really? I have no idea, but well, I mean, let me tell you this. So originally, friends of mine had a company, what was it called, Cyclocell. And I knew they were in trouble when they said we're going to use roscovitin to cure cancer because, that just inhibited all CDKs, and there was no reason to think that that was going to do anything at all. And then I heard Laurent Mayer give a talk how he explained how actually you didn't want specific CDK inhibitors. The whole idea was to have them with a sort of carefully tailored, constellation of cross-target effects, because that was what was actually effective. So, and what was it? There was one of these things. I can't remember what it was. It was it's gone from me now. There was one other one that showed some promise, and it turned out it was affecting transcription because it was inhibiting the CTD kinase.
So even the idea that inhibitory RNAs do the same effect doesn't discourage you from thinking it's still off target?
No, because inhibitory RNAs also have off-target effects.
There you go.
I'm a great believer in highly specific knockouts. Okay? Yeah. So if you could sort of take a cancer cell line which was relying on CDK4 and 6, and you could CRISPR out CDK4 and 6, and then they stopped growing, then I think I would believe it. Okay. I think there would be no alternative but to believe it. Okay? But it could still, of course, be that CDK4 and CDK6 are actually doing something other than phosphorylating RB. That's another article of faith, which I really don't know how well substantiated that is. You can see I'm very skeptical. We were taught, when I was an undergraduate, the professor of biochemistry said, don't believe what you read, especially if it's in a review. And I think that's pretty good advice, actually. In general, the primary literature is true, but the interpretation, of course, the data are right, but the interpretation is the thing you have to question.
Yeah, there's no data. There's just.
And so it slowed us down quite a lot. So we always wanted to find out things for ourselves, and I think that's a pretty healthy thing. I mean, yesterday we were discussing whether big data was a good idea or not. And I was really amused. I told the story, which I found out by more or less by accident. Somebody told me about it. Where, do you remember there was it was the ENCODE project, and they discovered that mouse liver looked much more like mouse than liver, if you see what I mean. I mean, in other words, you got all the messenger RNAs out of human liver and mouse liver, and they weren't terribly similar, okay, which was a bit surprising. But they were very similar to the other human constellation of organs and RNAs. Front page news in Nature. Mislabeling of the database. Completely wrong. Absolutely bogus. You know, somebody just put the wrong tag on all of these genes. And that was what explained this surprising result. When you put the right tag on, surprise, surprise, liver looks like liver, whether it's a mouse.
Yeah, if it sounds too good to be true.
Yeah, you know, but, and that's big data for you. I mean, you don't know how good even the original data was. You know, did they make good messenger RNA even? You have no idea, actually. No idea at all. You know, there's no... In my day, you could tell that because you could see whether the RNA was degraded or not. But nowadays, you just put in a kit or even a machine. You know, you add it to your beige box, and out comes this gigabytes, terabytes of big data.
Yeah. So the definition of big data is data without algorithm? Is that...
Well, not without algorithm, but without much quality control. I think. So I'm a great believer in small science. I mean, don't get me wrong. I think the sequencing of tumor DNA genomes has been absolutely fascinating, and led to but it's only made the problem much worse, of course. Now we know the astounding heterogeneity of these things. But that's the good news is that there's tons and tons and tons of stuff to be found out still. You think we're so smart, but actually we barely scratched the surface, I think.
Yeah.
Don't you?
That's true. Yeah. Let's see whether there are other questions.
Yeah.
Everything ends.
Oh, there's one. Right in the middle, right at the back.
There's a microphone running towards
coming, and I'm coming too, because I like to get because I like to see who I'm talking to, and my eyesight is so terrible.
You're following me.
Yeah. But he has to talk.
Yeah.
So,
Sir.
I have a question. How did you find the balance during your career between doing research that would be funded and doing research that really interested you? How do you walk that tightrope?
Well, I guess I was very lucky, and we never had very much money, at least to begin with. So, for example, all the work in Woods Hole had no funding at all. Well, that's not quite true. The courses had some NIH funding. And I got, S35 methionine was provided by Amersham International because a guy called Mike Brady had consulted with us, and he was a very nice man and generous, and it didn't cost him much to send us 5 millicuries of S35 methionine. So that was really good, actually. I always didn't like it when I had some big grant, because then you had to sort of felt a certain responsibility to spend it responsibly, and moreover to sort of do what you said you were going to do. And that's not always a good idea, actually. So it's a tough one, that. I mean, if you get a huge grant, then you have a heavy responsibility to the granting agency. So there's a lot to be said for having a sort of private income of your own and just doing whatever you like to do. And I think, I can't tell you how to do that, but the more you can kind of do that, the better off you are. And I don't know whether, it's an interesting question whether things have changed. I mean, at the time I had friends who had these sort of big program grants and things, and it always seemed to me they didn't do really as well. The program grants were largely wasted. We used to subsist on little tiny project grants, and it was good because, you'd apply for a project grant, then you'd find something out, publish a nice paper based on that, and of course they'd give you the next one. So you sort of went in 3 or 5 year steps forward, and it seemed to be completely fair. If you do well and find stuff out, then you get the next grant, and if you don't, you probably won't. So it's always nervy, especially when you get to grant. It's tough, but that seemed a much more honest way of going. But I've then discovered that granting agencies don't like that at all, because what they like is to give the Grand Panjandrum, an enormous grant, which the Grand Panjandrum then distributes according to his whim or her whim, right? And I don't think that's such a good way of doing it. I'm still rather a believer in, small. I mean, of course, if you're sequencing the human genome, you have to have an organization and all that. But many, many things, I think, still can be done with relatively modest resources, although, the fancy microscopes that people like to use nowadays are expensive, so you need to persuade. But, probably you've got a friend who has one that they don't use all that much. Whew! God.
Yeah.
Anybody else back here? Oh, yes, look, here's one. Really close.
Okay, have there been any attempts to identify the off-target effects, the potential ones, by means of using global kinome profiling via peptide arrays so far?
I don't know, actually. I'm pretty ignorant. I was very impressed. I've heard Dennis Sleman give a talk, and it was a very, very nice talk. He's a good scientist. So, yeah. Don't tell him I think it's an off-target effect. I don't, what do I know? But whenever something doesn't make sense, you're in with a chance. You ought to consider the possibility that it's not working quite the way you thought it was working. It seems to me there's something slightly, why would these particular cells be uniquely sensitive? And probably they're dying. Is he triggering apoptosis in those cells? And if so, why and how? That's, that, as you know, it's a very complicated business. And sometimes you have a pretty good idea, but often you don't really.
Tim, there's a young fellow down here who has a career question.
Okay, let's. I'll go down, okay.
Tim, just to share one of my problems with you and with your presentations, when a protein would be negative, being there, being not there, active, not active, I'm surprised. Whatever I looked into the effect and also looked into pellets, other statuses, scaffold-bound complexes,
yes,
etc. So a protein where you now suggest its minus is not there, degraded or what, is very often there, but in a different form, in another fraction, etc. Did you control this for you?
This is pretty well controlled. No, it really has gone away. I mean, it really isn't there. It's chewed to pieces by, the, what you call it, the proteasome.
So it's degraded.
Yeah, it's degraded. And the way the experiments were done, actually, we had all the, not only did I suck up the eggs, but I also sucked up the seawater. So if it wasn't there, something had happened to it. I mean, of course, it might have. If it got polyubiquitinated, it might have disappeared up the gel. In fact, that was, to some extent, the case. But there is really a protease that gets rid of it.
Just as you had examples of oocytes and eggs, and thinking of John Gurn's Xenopus in particular, the great biological problem, which tells you a lot, is, say, take the early stages after fertilization, say in a frog, like Xenopus. You have abundance of proteins there.
Yes. I mean, you have an incredible stockpile of everything you need, right, basically.
In other complexes, though, they are inactive, they don't...
Yeah. I mean, there are things like annular lamellae. I love annular lamellae.
Structure, and they're somewhere else. They are even sometimes in the nucleus somewhere else. So one and the same protein you think is not there, but it may be there in a completely different organization and complex arrangement.
Yeah, but I was just adding SDS and trichloroacetic acid, I mean, you sample absolutely everything, and it certainly wasn't there anymore.
The total?
In the total, yeah. And in fact, you can prove that when you have antibodies against it. It really does go away. Believe me, it goes away.
Any more questions?
Anybody else?
Oh, there's one up there.
Aha. All right.
Hey, well, thanks for the nice lecture. I really enjoyed this. This is more a question about work-life balance. And this is about this. For me, it seems that in the northern European countries, for example, you have this scientists often work a very strict 9-to-5 schedule with sometimes, I mean, just a personal observation.
9 to 5? No.
With sometimes sending and working from home and so on, in the evening. But then in the US, what I hear from friends there, it seems like they're supposed to be in the lab all the time, including weekends.
Yes.
Well, in Germany, for example, this would not be the case.
Oh, really?
Expected of you.
Oh, well, that's a serious... I always thought, when I went to the Pasteur to give a talk one time, and they had this fabulous lunch with wine, and I realized that the lunch was the great enemy of French science. The lab was a total life support system. I mean, I was normally there until 10 o'clock every night, sometimes till 2 o'clock. And I certainly, we always worked on Saturdays until, well, usually there was a program on the radio called Jazz Record Request, which usually marked the end of the day at about 6 p.m. on a Saturday. And we'd always come in on Sundays because you didn't, set up an overnight. So, and I never took any holidays either. But I went to a lot of meetings which were just as good as holidays, right? I mean, that's fine because you meet new people and you go to interesting places. And so I think, well, put it this way, I think there are some people who are incredibly well disciplined and can really do stuff on a basis, but I couldn't. I just had to be there. I regarded the lab as a total life support system. And I liked my lab mates, and we.
So you would expect this of your PhDs? That
I was always surprised when I went to Clare Hall that there were so few people there on a Saturday morning. That wasn't what I was used to. But one Spanish postdoc realized that I was always there on a Saturday morning, and we had our best conversations on Saturday morning because the phone didn't ring and there were no meetings or anything like that. So that was very good. And in Woods Hole, there was a lecture, I think, at 9 a.m., and I was usually working with the kids until about 2 a.m. We did actually sort of suffer from sleepless burnout in the end, because you just couldn't keep that regime up indefinitely. You do need some sleep. But very intense, I mean, I like that. I like that. So I think this question of sort of work-life balance, I think it's pretty... Because you are as you rightly point out, you are in competition with people. If people work more and longer, they're going to find out more faster.
Well, the question is if they're still effective, I mean...
I think they are. I'm sorry to say. Hate to tell you. Because they're sort of more immersed in it, I mean, it's just... when you're really on the track of something, it's absolutely obsessional. In my experience. Now, of course, there are long periods when you're not on the track of things. You're very depressed, and you can barely get up the will to go into the lab at all. So, you have your ups and your downs. Right? I mean. And, an awful lot of the time, things aren't working properly. You're not really on the track of anything. And I've more than once in my life had the feeling that it was time to give up, and I never find out anything interesting again. But you just got to sort of struggle to keep going. But it's such fun when it's going well. I mean, it's indescribably much fun. To know that you're the first person to really understand something that everybody would like to understand is an absolutely thrilling feeling, like nothing else.
I think if our head of administration hears this, he's going to wonder about hiring a British cook. If there are no longer any questions, I would like to once again thank you very much for a wonderful lecture.
Thank you for having me.